Welcome back, everyone. Today we're taking a deep dive into a particularly fascinating case report, canine olfactory ganglioneuroblastoma. Buckle up, because we're about to unravel the complexities of this rare tumor. And trust me, it's not just a mouthful to say, it presents some really unique diagnostic challenges too.
Oh, absolutely. These tumors, even for us in the field, are super intriguing.
So this case study that we're diving into, it's all about this 14-year-old miniature schnauzer, right? And where the poor guy was showing, the classic signs, nasal tumor symptoms, congestion, bleeding, sneezing, discharge, the whole works. And it just wasn't getting better with, the usual treatments, antibiotics, anti-inflammatories.
Yeah. And when you see those kind of persistent symptoms, it's a huge red flag, right? You know something else is going on.
Right. And then they did a CT scan and lo and behold, there's a mass right there in the nasal cavity. But here's the kicker. There was already bone destruction. And that really pointed towards an aggressive, invasive kind of tumor.
Oh, for sure. I mean, bone destruction, that's serious stuff. It's not just growing. It's like eating away at the surrounding tissue.
Exactly. So they needed to figure out what they were dealing with. So aspiration biopsy, that was the next step. And the results, well, they kind of took them down this rabbit hole into the world of peripheral neuroblastic tumors, PNTs for short.
Yeah.
And these tumors, they can come in a range of forms, from like the less mature neuroblastoma to the mature ganglioneuroma.
Yeah.
And then you've got ganglioneuroblastoma, which kind of sits right there in the middle.
It's like a spectrum, almost like a developmental spectrum within these tumor types.
Right, exactly. And what's really interesting about this is this specific type, canine nasal ganglioneuroblastoma, super rare, like barely any documented cases in the veterinary literature.
Yeah.
So, they're already breaking ground here. And to really get to the bottom of this, the researchers, they compared the schnauzer's samples to a whole bunch of different tissues.
Oh, yeah, they did. They went all out. They looked at normal olfactory neuroepithelia, which, for those who don't know, is like the tissue lining the nasal cavity. They wanted to see how those tumor cells stacked up against the healthy ones.
Right.
And then they went even further. They compared it to samples from other canine olfactory neuroblastomas and even threw in a spinal neuroblastoma from a different dog. So side-by-side comparison, super important here, figuring out what makes this tumor tick.
And the tools, they were using both traditional histology, the good old microscope, examining those cells, and immunohistochemistry, right? Which is like next level stuff. It helps you identify specific proteins within the cells.
Think of it like this. Immunohistochemistry is like a molecular spotlight.
Okay.
So you've got these antibodies that latch onto specific proteins, right, in the cells. And it's kind of like highlighting the VIPs, the key players in that cellular world. And for this case, it really helps them map out all the different cell types in the tumor and figure out their roles, like really intricate stuff.
Yeah. So let's get down to the nitty-gritty.
Yeah.
When they put that tumor under the microscope, what do they see?
Okay, so the tumor cells themselves, they had this kind of polygonal shape, and they were all surrounded by something called Schwannian stroma, which is, essentially like a support network of cells. But in this case, it had this really distinct look to it, very fibrillar and stained super intensely with eosin.
Interesting.
Right. And then, nestled within the stroma, they found these little clusters of neuroblast-like cells, which are, essentially immature nerve cells. And get this, the report even mentions some of these cells were arranged in these rosettes.
Rosettes?
Yeah, little circular formations of cells.
Okay.
And that can actually be a hallmark of certain tumor types, So again, another piece of the puzzle.
Right. And they also looked at the mitotic count, which tells us how quickly the tumor cells are dividing, right?
Exactly.
Potentially spreading.
Higher the mitotic count typically means more aggressive tumor. But here's the thing, in this case, it was surprisingly low, which is kind of weird because remember that bone destruction doesn't quite add up, right?
It's a real head scratcher.
Yeah.
Less aggressive mitotic count, but yet there's that damage.
That's exactly why this case is so unique and why it's so important to, to really dig deep into these rare tumors. There's more going on than meets the eye.
For sure. And speaking of digging deep, there's this table in the report, table 2 to be precise, that lays out the immunohistochemistry results.
Oh, yeah.
It's pretty dense, but can you break down some of the key findings?
Absolutely. So one of the most striking things was that the ganglion cell-like tumor cells, they tested super strongly positive for tyrosine hydroxylase.
Tyrosine hydroxylase. Remind me what that means again.
Sure. So it's an enzyme, a key player in making dopamine and norepinephrine, neurotransmitters that are usually associated with the sympathetic nervous system.
So finding this enzyme in these tumor cells, that's kind of unexpected.
Definitely. I mean, the olfactory system where this tumor started, it's not exactly part of the sympathetic nervous system.
Right. Intriguing. And the neuroblast-like cells, what about their reaction to this marker?
Didn't react at all. So right there, you've got two distinct populations within the same tumor.
Wow. Okay, so two different cell populations hanging out together in this tumor. What's that? Tell us about how it developed. Where did these cell types come from?
That's the burning question, isn't it? And to answer it, we got to dive into the possible origins, the neural crest versus the olfactory placode. And that's where this case really starts challenging, the traditional ideas about these tumors.
All right, so origins and what it all means. Let's dive in. Stay with us as we break it all down.
So just to back up a bit, in humans, these peripheral neuroblastic tumors, they're generally thought to come from the neural crest, which is this group of cells that kind of migrate all over the body during, embryonic development.
Right.
And they give rise to a whole bunch of tissues, including parts of the nervous system. But what's so interesting about this case is you've got these sympathetic nervous system-like cells, which, like we said, are derived from that neural crest. But then you also have those olfactory neuroepithelium-like cells, which have a more complicated origin, shall we say?
Oh, okay.
So the olfactory neuroepithelium, it actually gets contributions from both the neural crest and this other embryonic structure called the olfactory placode.
Interesting.
So having both of these cell types in the same tumor, like in the schnauzer, it raises some really interesting questions.
Right. Like, are we even sure how these tumors develop? It's like finding puzzle pieces from two different boxes.
Exactly. And the researchers, they actually propose that the neural crest-derived cells in this tumor, they might have kept the ability to differentiate into both of those cell types.
So they're saying these cells have some flexibility?
Yeah, like a plasticity that we wouldn't normally expect in a tumor.
Wow, that's fascinating. And this is backed up by the presence of that marker, SOX2, right? The one we saw in the immunohistochemistry results.
Yep, that's right.
Can you remind us what that marker means again?
Of course. So SOX2, it's a transcription factor, meaning it helps control the expression of other genes.
Right.
And it's known to be involved in keeping that stemness of cells, that ability to self-renew and differentiate into different cell types.
So finding SOX2 in the tumor cells means they might still have that early developmental potential, like they haven't fully decided what they want to be.
Exactly. And what's even cooler is that SOX2 is specifically linked to those globose basal cells.
Oh, right. The multipotent ones, the ones that could be the common ancestor for both of the cell populations in the tumor.
Precisely. We're talking about a tumor that might come from cells that could become a bunch of different things.
That's wild. So this brings us to the real heart of the issue here, this incredible plasticity of cells and the possibility of these unexpected developmental pathways in canine tumors.
Right. This case just opens up all these new ways of thinking about how these tumors start and how they behave.
Makes you wonder if this is something common in canine olfactory ganglioneuroblastoma. More research is definitely needed.
Oh, absolutely. But even with just this one case, it's incredibly valuable, challenging our assumptions, showing us just how complex these tumors can be.
Yeah, it really makes you think maybe there are other cases out there undiagnosed, just because we haven't been looking for this level of complexity at the cellular level.
It's a good reminder that science is always evolving. One unexpected finding can completely change how we understand things.
That's so true. This case just emphasizes how important it is to stay curious and open to new possibilities.
Couldn't agree more. As we wrap up this deep dive, I keep coming back to that last sentence in the report.
Oh, yeah.
It's like they're throwing down the gauntlet, a challenge to all of us in the field.
Which one are you talking about?
The very last one. It says, further research with more cases is required to determine whether this is a common feature of canine olfactory ganglioneuroblastoma.
Oh, yeah. Powerful statement for just one sentence.
Right. It's like they're saying, hey, we've just scratched the surface here. There's so much more to learn. We need to investigate if these findings are, just a fluke or if they represent something bigger.
Definitely highlights that Even with all we know in veterinary medicine, we still have a lot to learn about these tumors.
Oh, absolutely. And this single case, it's like it's opened up a whole new world of questions.
This case has been mind-blowing, that's for sure. It challenges everything we thought we knew about these tumors, where they come from, how they develop.
Exactly. And it all points back to this amazing plasticity of cells, their ability to kind of break the rules and deviate from those expected developmental pathways.
It makes you wonder, what does this mean for how we understand and treat canine tumors in the future? I mean, are we missing other cases just because we don't expect this level of complexity?
That's the million-dollar question, isn't it? Could this change how we diagnose and treat these types of tumors?
Wow, so much to consider. But for all the vets and vet students listening, what are the main takeaways from all this?
Well, first off, I think it's a huge reminder to keep an open mind, right? When you're dealing with nasal tumors in dogs, especially, consider those rare diagnoses like olfactory ganglioneuroblastoma.
Yeah, and it really shows how important it is to use all the tools we have, like traditional histology, along with these more advanced techniques like immunohistochemistry.
Exactly. You got to get the full picture, right? And maybe most importantly, this case shows us that we're still learning so much about canine tumors, their origins, their development. It's all still being explored.
This deep dive has been a wild ride. Canine olfactory ganglioneuroblastoma, who knew it would be so complex?
Right. It just goes to show, in science, there's always something new to discover. We're just scratching the surface.
Well, that's about it for today's deep dive. Thanks for joining us as we explored this fascinating case. And from Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.